Hello, everyone. I'm Max Nomura from Asakase Corporation, Japanese chemical and material companies, and work as the chair of International Affairs Committee of Pharm for Innovative Regenerative Medicine. It's my great pleasure to have opportunity to organize this Pharm's workshop about our activity and current progress or regulatory harmonization. The name is APACRM. The APACRM, Asia Partnership Conference of Regenerative Medicine, aims at harmonizing and optimizing regulations among Asian countries and regions. Pharm has organized APACRM, an annual conference since 2018, and has closely collaborated with industry groups such as the China Medicinal Biotech Association, CMBA in China, and the Association of Biotechnology Led Enterprises, AAPI in India, a council for Advanced Regenerative Medicine, CARM, and the Strategic Center for Regenerative Medicine, SCRM in Korea, Singapore Association of Pharmaceutical Industrial, SAPI, and Biotechnology and Pharmaceutical Industries Promotion Office, BPIPO in Taiwan. Our latest APACRM focused on dialogue with regulators based on survey results from 3 working groups: non-clinical, quality assessment, and environment impact assessment setting for cell and gene therapy product. Through panel discussion comprised of major Asian regulatory agencies, regulation differences and their underlying rationales had been more clearly defined among the participating country and region. The hope is that the environment of harmonization is cultivated by sharing experience regarding product and development, and by sharing experience regarding product, and opinion and regulatory guideline. Moreover, more proactive effort on regulatory innovation, for example, necessity of a new guideline regarding regenerative medicinal product can assist with the regulatory harmonization and optimization across Asia. This would enable the rapid delivery of CGTs to more patients, thus achieving market maximization of CGTs throughout Asia. This session will explore prospects and challenges to promote harmonization and optimization regulations on regenerative medicine product among Asian countries and regions. We have 4 speakers today. Firstly, Hiro will make a presentation about recent achievement on APACRM Working Group 1 for non-clinical assessment of cell therapy product. Secondly, Ryan will explain the result from Working Group 2 for the regulations in Asia on biological ancillary materials. Finally, Miyano-san and Yasumoto-san will introduce a starting point of Working Group 3 for environmental impact assessment of cell and gene therapy product. Okay, Hiro, please go ahead.
Hello, I'm Hiroshi Karasawa. On behalf of APACRM Working Group 1, I will talk about the dialogue between regulatory agencies and industries on non-clinical assessment of CAR-T products. Here is my COI declaration. I'm a scientist employed by Japanese pharmaceutical company, Daiichi Sankyo. Here is disclaimer. Here shows contents of my talk. First, I briefly review the APACRM and our working group activity. Next, explain specific aims and scope of Working Group 1 in 5th APACRM. We have talked with Asian regulators in APACRM in two different perspectives. In M1 part, we discussed about non-clinical package of conventional CAR-T products targeting hematological malignancies, such as anti-CD19 CAR-T products. In M2 part, we talked about potential issues in non-clinical evaluation of next-generation CAR-T products. Today, I will summarize our approach used in 5th APACRM and share the details of the discussion between regulatory and industry and some topics for M1 and M2 respectively. APACRM, Asia Partnership Conference for Regenerative Medicine, was first organized in 2018 by Asian industrial groups for regenerative medicines. Our mission is to support the development and delivery of high-quality regenerative medicine products to all patients within Asia. For this end, it is very important to promote regulatory alignment in Asia. Considering for the Asian harmonization in final, we are promoting the dialogue between regulatory agencies and industries. In the latest APACRM in this April, we invited regulators of India, Singapore, Taiwan, Korea, and Japan for panel discussion between regulatory agencies and industries. Now moving on working group 1. Among 3 working groups in APACRM, working group 1 is focusing on non-clinical. Our objectives are to compare the non-clinical assessments for regenerative medicine products in Asian countries, regions, and to extract regulatory scientific issues to be discussed with regulators, considering for the Asian harmonization in final. To this end, experts for non-clinical assessment of regenerative medicine from India, China, Korea, Taiwan, and Japan, shown here, have joined to the working group 1, and I, Hiroshi from Japan, is now leading the group. Here shows our previous works of working group 1 in the first term. These are good examples telling about our activities. In the first term, we focused on regenerative medicine products derived from MSCs, mesenchymal stem cells, and conducted comparison of regulations and required non-clinical packages among 5 Asian countries. Our survey results were published in the review article in Site Therapy, and the summary of discussion with Asian regulators in the last APACRM was published as a proceeding paper in the Site Therapy journal. Also, we have opportunities to present our works in several scientific meetings, including the last year Cell and Gene meeting on the MESA. From now, I'm going to talk about our second term activity focusing on CAR-T products. Our objective is to investigate the non-clinical assessment for CAR-T products in Asian countries and regions, considering for the future Asian harmonization of regulations in final. 38 experts from India, China, Taiwan, Korea, and Japan joined this activity. Considering for the vast diversity of current CAR-T products, we have set 2 specific aims. M1 is considering for the conventional CAR-T, such as an autologous anti-CD19 CAR-T, and to compare the non-clinical packages in Asian countries to extract discussion points. M2 is considering for the next generation CAR-T, adapting novel technologies to find the regulatory scientific issues for non-clinical evaluation of CAR-T products with such technologies. I will briefly show the summary of our works for each aim and share the points of discussion with Asian regulators in 5th APACRM. M1 is dealing with conventional CAR-T products. As you know, the development of CAR-T products targeting hematological malignancy is becoming active worldwide. Here, we have compared the non-clinical study packages among 5 Asian countries to extract regulatory scientific discussion points. To this end, we conducted a case study using a virtual CAR-T products. This means, first, we prepare a virtual CAR-T products for the case study and prepare the non-clinical study packages in each country region according to their local regulations, and compare the packages to find the key discussion points and discuss not only in industries, but also with regulatory agencies to propose the future perspectives. This slide shows details of our virtual CAR-T products using our case study. It is an autologous CDXX CAR-T targeting for B-cell malignancy. It has a typical second-generation CAR-T molecule shown here, and the whole product profile is very similar. It's very conventional. I think it is enough to imagine anti-CD19 CAR-T product such as Kymriah or Yescarta. Here summarizes survey results and key questions based on the survey. Overall, non-clinical packages prepared by 5 countries of Asia were basically similar, but we could extract some discussion points. In the 5th APACRM, we asked Asian regulators about 8 questions shown here and received valuable comments from regulators on each of them. Today, I will share the essence of the dialogue on these 3 topics, 3 shown in blue for pharmacology, CKBD, and safety. First, in pharmacology, we asked how to estimate the clinical dose. Through our survey, 2 different perspectives are elucidated. A, we can translate the optimal animal cell dose to the clinical cell dose. And B, we accept to follow or refer the previous clinical studies of other products with similar profile. Basically, all countries of industry accept B, but Taiwan and India required A also. So we asked regulators if the exploration from dosing to animal recommended to estimate the clinical cell dose. And here shows the answers from regulators. Regulators of India pointed the importance of estimation using animal dose. Japan PMDA pointed the difficulties because of GVHD response, which is inevitable in studies administering human T cells in animals. Next, the question is in CKBD is a bit interesting about the CKBD variation of CAR untransduced cells. When introducing CAR molecule using lentiviral vector, its efficiency is not 100%. Thus, CAR-T products will contain T cells that are not transduced CAR molecule. In the case of autologous CAR-T products, CAR untransduced cells are indistinguishable from patients' own T cells. Thus, it is reasonable that all regulators answered there is no need. However, in the case of allogeneic CAR-T products, CAR untransduced cells shown in a different color are not identical to patients' own T cells. To this case, all regulators again answered no. This question may be overthinking of us, industry side. Japan PMD clearly answered that unless there is a specific concern that affects CKBD, CKBD variation of CAR and transduced cell is not required for next generation CAR T cells as well. The next topic in safety is about the methods used in evaluation of target binding. Traditionally, tissue cross-reactivity TCL test based on immunohistochemistry is a gold standard to assess the binding potential of the binders. Recently, as a novel method, plasma membrane protein array PMPA has been developed for off-target binding assessment. This can assess the binding to more than 5,000 human plasma membrane protein using protein array. Our question to regulator was, when PMPA is conducted separately, is additional TCL test necessary to initiate the clinical study? Answers from regulators are shown here. Some consider the PMPA alone is sufficient, and some considered no. CDSCO India pointed out the purpose of the test to identify the site of on-target binding, which were not previously identified. Anyway, using virtual CAR-T products, we could extract discussion points for conventional CAR-T and have valuable input from regulatory agencies. From this slide, I will talk about AIM2 considering for the next generation CAR-T. Despite the great success in hematological malignancies, there exist critical limitations for current CAR-T therapy. Limitations are roughly classified into two groups. One, limitations derived from autologous therapy, like this, and two, limitations in efficacy and safety, particularly for solid tumor. To overcome these limitations, intense research and development are conducted as next generation CAR-T products. This cartoon summarizes the circumstances surrounding next generation CAR-T products. Based on these background, we have selected 4 topics shown here: allogeneic CAR-T, iPSC-derived CAR-T, CAR-T targeting solid tumor, and other components than CAR. And we extracted discussion points. Here shows the discussion points selected on each topic by industry. In the 5th APACRM, we asked Asian regulators about 7 questions shown here. Now, I will share the essence of dialogue on these 2 topics shown in blue, topics for CAR-T targeting solid tumor and other components than CAR. This is a topic about tumor microenvironment, TME. TME surrounding solid tumor is one of the most important factors affecting the efficacy of CAR-T cells. Here showed 3 available model. Here shows the 3 available options of the solid tumor model for in vivo pharmacology study. The first one, syngeneic model in which mouse tumor is transplanted to normal mice. It is ideal for reconstitute mouse TME. However, it's not for humans. The second one is PDX model. In this model, patient-derived tumor itself is transplanted to immunodeficient mice. Its TME does not include host immune system, but is considered more clinically relevant. The third one is CDX model. In this model, tumor cell line is established from human tumor is transplanted to mice. Its TME does not include hosting. During the establishing cell line, tumor cells lost its nature of their original tumor tissue. However, this model has been used in the field as a first choice because of its better availability and reproducibility. Our question to regulator is asking if pharmacological evaluation with CDX model alone is sufficient or not. To this question, regulators except for Japan answered no. But they did not suggest to use other two in vivo model. Rather, they suggested to include additional in vitro pharmacology data supporting the efficacy to solid tumor. This is the final topic I share today about the safety evaluation of CAR T cells that express ligand not cross-reacting to mice. This is the question using a case of next generation CAR T cells reported in Cancer Cell in 2015. This is promising CAR T cells that express 4-1BB ligand in addition to anti-CD19 CAR molecule. And the point is that this 4-1BB ligand does not cross-react to mouse 4-1BB receptor. We asked regulators the necessity for the separate in vivo tox study with 3 options here. However, all regulators except for India answered it is difficult to answer based on these limited information. This is a bit disappointing to us, but reminded us the importance of case-by-case principle in non-clinical evaluation of medical products. We need to clarify the issues and point more clearly for more fruitful discussion between regulatory agencies and industry. For closing remark, we really appreciate all the Asian regulators who joined 5th APACRM and involved in dialogue between regulatory agencies and industries. We hope this activity will accelerate the mutual understanding between regulatory agencies and industries and will lead to the Asian regulatory harmonization that will help the development and delivery of novel regenerative medicine products to all patients within Asia. Now we are preparing the manuscript of the proceeding paper with the 5th APACRM, and we hope details of our dialogue between the regulatory agencies and industry will be available soon. This is the end of my presentation. I really appreciate all the members involved in APACRM Working Group 1 and other persons and organizations, including firm and regulatory agencies for generous support for our activity. Thank you very much.
Okay, thank you for your good presentation and so informative presentation. Thank you. So I'm just quick question is that so is any plan to investigate more? So it's a CAR-T and conventional and advanced CAR-T about that's very informative. But is any plan to investment more about like that? Or next, what is next? Next is not CAR-T or not? So could you please explain the next plan?
Okay, thank you. Yeah, actually, I think we could do very good dialogue between regulatory agencies and industry in the last APACRM, and I think their comment is very proactive to very proactive. I think actually the discussion point in the previous meeting was very limited one. So I think we can do more about CAR-T. So now the working group 1 is continuing our activity about CAR-T to discuss more and extract more discussion points. And then we are planning to present them in the next APACRM, 6th APACRM.
Okay, good. Thank you. And so one more question. It's could you explain what is the impression point? I mean, that's maybe before this work, after this work, as your own impression, what information is impressive for you or not?
Yeah, that's a very difficult question. But during the question and answer, I felt that some of our industry question is maybe over consideration of our industry side. And also in another question, we felt that information is very limited and it doesn't work well for asking the comments from the regulator. So anyway, so for the future activity, we think more about the background of CAR-T and our products plan, and then asking the question to the regulator. That is very important, I think.
Okay, thank you. Thank you for that.
Thanks so much.
Yeah, thank you. So next is Ryan presented. So.
Hi, thank you everyone for being here this morning. And this is Ryan Chen of Espodia, and I'm very excited to participate in this panel and to present our group work about the regulatory differences in Asia for the use of biological and ancillary materials in cell and gene therapies. So before I start, I'd like to introduce you the background of this study. Asian Partnership Conference of Regenerative Medicine is a nonprofit annual event to bring Asian regulators and industries together and to help promoting mutual understandings of the standards in cell and gene therapies, and with benchmarks to our counterpart in North America and Europe. And this annual event is sponsored by Forum for Innovative Regenerative Medicine, aka FIRM in Japan. Now, just speaking from my own experiences about this mutual understandings, I sometimes complained privately why I needed to do more in my submission packet in one country if I have good compliances position in another. Yet through this group work over the past 2 years, I came to understand better why those additional work were required as such. Therefore, I can say that the works carried out by the Asian Partnership Conference have been bringing true values. It's like mapping a North Star to the cell and gene industry globally. Now, let's first look at the state of the industry since 2020. These two charts here showed active studies in terms of geographic distributions and stages of development. North America and Europe still dominant in terms of numbers of developments, and the regulations there continue to evolve along with the growth momentum. While Asia here, you can see that as a whole takes relatively less shares. When it comes to the stages of development on the right-hand side here, we can see that close to 80% are still in early stages, including both preclinical and phase 1s. And therefore, we believe that both the regulatory environments and the industry in Asia still have a very good chance to catch up in the international markets. I believe that the audiences here all know that developing cell and gene therapies is very hard, but from discoveries to scale up and to mass production is even harder. And our work group 2 is focusing on regulatory quality requirements and some critical areas surrounding the CMC or the life cycle from start to downstream manufacturing and to the final launch of the products for treating patients. And over the past 2 years, our work group investigated the eligibility of starting materials across Asian countries and has generated 2 publications, including a recent one published in April this year. And now, as a nature extension from our previous work, we further weighted in on biological ancillary materials due to its significant impact on both safety and efficacy, which is very profound right from the beginning phase of both research and production. So within the next 15 minutes, I will give you some rapid-fire snapshots of what we have found. And in the meantime, I encourage you to read our publications for more details. According to USP 1043, ancillary materials is defined as materials used in manufacturing processes, but not intended to be present in the final products. And as I just mentioned, the impact of ancillary materials on the quality, safety, and efficacy needs to be carefully assessed by the developers with the ultimate goal of patient protection. So the scope of our study looked at the Asian regulations on 2 subject areas. One is about the general risk assessment, and the other is specific to bovine serum prostaglandin, as well as recombinant proteins. The regulations we collected here include both issued guidelines and local pharmacopoeias. And through our study, we took 2 inseparable principles, including risk-based and the science-driven approach, to review the regulations in each country and to highlight their different rationales. And to our methods, it contains 2 sequential elements by first initiating a questionnaire for regulatory agencies to respond, and then to follow with roundtable discussions with all regulators sitting together on both questions and answers. And we expect that such a process shall bring the industry a more comprehensive understanding of what needed to be done to be in good compliances. And based on our surveys, this slide shows a quick overview of both general guidance on how to perform risk assessments, on the use of ancillary materials, as well as specific guidelines, regulations on certain materials used during the production processes. And in this table, we can see that comparing with the US and Europe, there do exist quite a gap among Asian countries in the developed guidelines on ancillary materials. But this slide gives a much more detailed, a comprehensive review of guidelines classified as general and materials from human origin, animal origin, and those used to human or animal sourced materials in the substance production, like recombinant proteins. And instead of giving you a detailed account line by line in this table, here is a summary of our findings in the first step of our study, and 2 points stands out. First, all the available guidelines are very limited to general and to a few materials. And also, there are very clear differences in each country or region. And second point, when it comes to specific ancillary materials, there are different requirements in each country. But anyhow, we did find a silver lining here is that some regulatory agencies in Asia do reference the published guidelines in the US or EU. In our second step of our investigation, and based upon the survey results, we held a roundtable discussion on April 7th this year. The discussion agenda was designed around 4 topics, seeking comments from each of the participating regulators from Japan, Singapore, India, Korea, Taiwan, and Malaysia. And we also invited an expert, Dr. Yuji Sato, to discuss from the perspective of regulatory sciences. And these 4 topics include overviews of relevant regulations, performing risk assessment, using licensed commercial drugs as ancillary materials, and finally, their suggested points to consider for specific materials in production. And in the following slide, I will provide some highlights of the discussions that day. So by reviewing the relevant regulations, this slide summarizes our questions with comments from each of the regulators. And I just wanted to point out several factors for the audience to consider. In Japan, when it comes to sufficiency of foreign certificates here, it is essential, based on comments from the regulators from Japan, it is essential to follow the local standards for biological materials and their subchapters on materials of human, ruminant, and animals. While in other countries, foreign certificates like the certificates of suitability from EDQM can be accepted as proofs of safety. And in this discussion, we also asked about what are the common problems and the best practices based on their review experiences. And all of the guests suggested the one point, which is avoidance of animal and human-derived materials is the best for every developers to pursue. And our second topic of panel discussions was about performing risk assessments. As a general guidance in our survey, all the risks can be classified in 4 categories with references from USP 1043, EP 5212, and China Pharmacology, where risk levels is considered having the highest risk. So it is important for the developers to really look closely on the biological origins, documenting the traceability and the production steps to control, to inactivate, or to remove the ancillary materials. And also, it is very important to present the rational and scientific qualifications program for regulatory reviews. Then based on the risk classifications just mentioned, we further asked the regulators 2 questions on conducting an assessment. Okay, and 2 points to share here. First is to pay attention to the risk of the adventitious agents like HIV, viruses, and the bacteria in the materials used. And with regard to the substance of human and animal origins, this should be tested for the transmittable pathogens and their potential immunogenic responses to the patients. And second, the developers should present evidence on at least both the primary and secondary components used in the substance for producing the ancillary materials. Here I'll give you a rule of thumb from the various comments. It is about really take a risk-based approach of all potential risk exposures that could affect the safety to patients. Our third topic to the regulators was about using commercial drugs approved in other countries as ancillary materials. And it is generally considered that approved commercial drugs have lower risk for CGT production because the commercial drugs are manufactured under appropriate GMP controls and approved for their safety and quality by regulatory agencies. Still, there's no guarantee. Let me repeat again, there's no guarantee in the reviews of some countries due to those licensed drugs' original intended use and their risk-benefit profiles for those drugs. As a result, we still recommend engaging with the regulators as early as possible to avoid any wrong presumptions. Our final topic for discussion was about the regulations on some specific materials, including bovine serum and prostitrypsin. And here is a list of all the available guidelines we have identified. As you can see from this slide, for Asian countries, relevant guidelines are very limited and are mostly present either in local standards or pharmacopoeias. Yet for some other countries, they do fall back to the standards from Europe or the US. And this is summarized in our next slide. In addition to general assessment for specific animal and ruminant-derived TSC relevant ancillary materials, we asked the regulator if there are certain points to be considered for the developers and the manufacturers. And given the fact that both bovine serum and trypsin are very common animal-derived materials, Singapore, Taiwan, Korea, and India all accept ETQM certificates and put particular emphasis on the country of animal origins, the residual testing data. And Japan, on the other hand, could take ETQM certificates as references, but still require presenting extensive testing data. For example, amount, when in the process those materials are used, and the methods of use to prove the safety. So I also want to bring your attention to the country-specific differences here, like for Malaysia as an Islamic state. There are specific requirements to test on traces porcine and clear label declaration for the materials. In closing, if we could give you an overall observations and suggestions from our investigations and the dialogues with the regulators. Because concerns about risk of ancillary materials do vary country by country. For example, TSC risks contamination of prevalent viruses in some countries. Each country or region may have different speed to issue or revise their guidelines depending on their accumulated experiences. Therefore, the risk assessment approach, considering the biological origin, traceability, the production steps, and the quality control over the manufacturing process, become even more important. Meanwhile, we do recommend moving towards defined serum-free media and recombinant trypsin in order to reduce the risks from bovine serum and porcine trypsin. In the same time, to simplify the risk assessment while confirming an impact on quality, efficacy, safety of the final product. As a final suggestion to the audience here is for developers and the manufacturers to really start with the end in mind. That is, to take rigorous quality framework right from the beginning. The later you find problems, the harder it will be to make changes given the time and cost of delaying along with the life cycle of development. We would like to pay our tributes to those participating agencies for their openness and the many good advice, which include Singapore, Taiwan, India, Japan, Korea, Malaysia. Also, Dr. Yuji Sato presented as expert on regulatory sciences. Also, we want to acknowledge the contributions from our group members to produce this very valuable work of studies. And thank you very much for listening.
Thank you for your good presentation, a very comprehensive presentation, and to clarify the survey result. So finally, you summarized that we couldn't find no common requirement across a surveyed country and region. I understand that it's a difficult way, but I would like to know your comment, your feelings. How should we find a way to harmonize it on ancillary materials?
Okay.
Difficult question.
I think when we talk about harmonization, and to me, it's not about setting up common standards for all to follow, but rather to seek commonalities among all countries or regions and respect individual differences at the same time. So in the case of regulating ancillary materials, from our study, we see the common perspectives of risk assessments driven by science and data.
Yeah.
And on the other end, from our dialogues with the regulators, we also understand their genuine concern for the sake of patient safety. So to me, it's really not a conflict, but it's what's really needs to be done to make efforts. I believe this is where we, like the Asian Partnership Platform, could work on further finding the balances and really bridging multiple sides, including the regulators, industries, and the industrial standard groups together. Okay, and if we can come closer before we can enrich the regulations in both their efficiency and effectiveness, I think we are moving along with the journey to harmonize the standards. Yeah. And also, I think that if I may, there's an industrial group like ISO. They are really creating compiling good references. For example, in the case of ancillary materials, they have put together in their ISO 2399 general requirements for ancillary materials and the best practices for suppliers and users in using ancillary materials. Yeah. Okay, so all these are trying to minimize, all the efforts are trying to minimize the redundancy, at the same time to create because cell and gene therapies are still burgeoning and the emerging areas with a lot more to come. If we continue to accumulate our experiences, including with the regulators, I believe that there's really a true need that will drive the change and towards harmonization. So this is just my personal view on this.
Thank you. Thank you. So good suggestion. Yeah, maybe take time, much time to also complete it, but we'd like to do so. Such kind. Yeah. Thank you. So final question is, what's the next investigation items on working group 2? What's the next?
Well, I cannot say on behalf on our work group 2, but we are still discussing internally. But I believe, as I just mentioned, working group 2 is focusing on regulatory quality requirements throughout the life cycle, or CMC, from start to final launch. So this is some in between. We'll definitely find something meaningful and having the strong impacts for the global industry and the regulators. Thank you.
Okay, good. Very good. Thank you. Thank you so much.
Thanks.
Okay, hello. I'm Masamiyano from Chugai Pharmaceutical, Japan, and I'm happy to give you a presentation from our working group 3. Working group 3 is a new working group in APACRM, and we started about 1 year ago. And Mr. Yasumoto and I will show you the current status and activities of our working group 3. So this is the purpose and background of our working group 3. So now about 200 organizations join the Convention on Biological Diversity in the world. On the other hand, the implementation status of Cartagena protocol is different in each area or country. So we started working group 3 to discuss if we can harmonize or standardize some item of regulation related to environmental impact assessment or EIA in clinical or regulatory perspective. As a starting point, we started comparing the regulation regarding EIA in APAC area. The purpose of our survey is to compare regulation and understand the regulatory system related to gene therapies development in APAC area. And we would like to find common points and different points. We think that such analysis will give us idea how to collaborate together and facilitate efficient development of gene therapy products in APAC area. So this slide shows the comparison of regulation, our survey results. And I will not dig into details, but we surveyed the status of implementation of Cartagena protocol and domestic regulation and health authority review system in each country or region. At the APACRM this year, regulator pointed out to difficulty of the harmonization of such regulation regarding EIA or Cartagena protocol. But on the other hand, he also mentioned that this kind of table will give us, a really clear idea and very informative and valuable table. I will hand over to Mr. Yasumoto.
Yeah, thank you very much. Yeah, please let me explain from this section. My name is Masahiko Yasumoto, working for Non-Carrier Corporation in Japan. My topic covers the ongoing discussion in Working Group 3 after APACRM conference. Please go to the next slide. At the APACRM meeting, I proposed how we, Working Group 3, established the next discussion according to this slide. In this term, I wanted to focus on more specific and detailed discussion based on 3 points. Number 1, discussion covers not only environmental impact assessment regulation, but also the other usual regulations. This is expressed as scientific regulation. Number 2, in the definition of gene therapy, I wanted to narrow down the subject materials to be in vivo gene therapy instead of in vitro. I wanted to use a certain mock genetically modified organisms as a common tool for our consideration. And number 3, since it was considered to be difficult to cover all the regulations and environmental impact assessment regulation throughout the entire clinical development, I proposed to set up a common background for this investigation divided into 5 segments: before clinical trial, at the same time of CTA and IND, and in parallel with clinical trials, regulatory consultation, and during marketing authorization. Based on these 3 points, I requested the APAC members for their opinion and acceptability on my proposal as a questionnaire. That's in the next slide. Yep. Yeah, please excuse me, this is a quite busy slide. This is the result of a questionnaire. To get the consensus with APAC members regarding my proposal, I prepared and delivered a questionnaire including these 4 questions. I would like to explain the result or responses in this questionnaire in the next slide. Yep. Yeah. According to the result of questionnaire, final consensus with APAC members is summarized in this slide. 1, mock GMO, adeno-associated virus vector. if additional specifications like preventive use or serotypes are necessary, we'd like to discuss more. And second, development stage. And from the very beginning of starting point of development to make submission of CTA or IND. Third, investigation items. All the necessary items required by your regulations, including environmental impact assessment. APAC members are currently correcting their regulatory frameworks based on these common background and tools. Next slide. According to the preferred background, like a mock GMO, development stage, and investigation items, we are requesting for summary presentations in each country and regions covering regulatory requirements to submit a CTA, IND to the agency, as well as environmental impact assessment requirement. After receiving such a summary presentation from the members, we will analyze and find out the similarities and the differences between country and the regions. Go next slide. This contains the example of TOC for such a summary presentation for your information. That is all. Working group 3 presentations. Finally, as mentioned before, this working group 3 was newly established last year to discuss the topic of environmental impact assessment among the APAC countries and regions. This topic is relatively difficult to discuss in terms of harmonization of regulation between countries and regions, because one of the reasons is that GMO are not only a therapeutic product, but also plant and animal and food, having a very wide-ranging definition. We should avoid the duplication of environmental impact assessment data as much as possible, and minimize the requirement just for therapeutic use and clinical use. So if we could use the same data internationally, we could deliver beneficial treatment to the patient earlier than ever. As a first of our approach, we would like to start a small investigation in Asia. And your cooperation of ARM and the other sponsored industries here is highly appreciated to extend our discussion. Thank you for your attention.
Okay, thank you for presentation. As I have a quick question about the maybe Yasumoto-san. So you mentioned about the mock kind of example product image of the. Could you explain more? So is it just adenovirus vector, or also something to cells, or not? So could you explain the more image on mock product image?
Thank you very much for your question. As I mentioned, that mock product would be adeno-associated virus vector. As you know, a lot of gene therapy products are currently being developed around the world. We are focusing on the adeno-associated virus vector as one of representative virus vector to correct the information on necessary regulation system and the regulation for the development from each Asia country and region.
Okay, I got it. Next question is maybe Miyano-san. So you presented about the survey results or questionnaires results, the number, counting number. So is any could you any findings about the country or region specific question or not? So some differences between the countries and region, among the countries and region, or not?
Thank you for asking. Yeah, so yeah, we looked over the table, and we didn't see any clear trend, yeah, depending on the country or regions. But so but this is like I said mentioned. So this kind of summarized comparing table will give us some idea what kind of barrier we can find in what country or region, and what point is similar each other. So like Yasumoto-san mentioned, we can use this kind of table to facilitate our development, earlier development in APAC area, we believe.
Okay, thank you. So as you mentioned about the summary presentation, so when do you intend to complete its presentation?
Yeah, we are thinking about we will present the summary table from each country's information at the APACRM next year. It is planned, I think, planned in April 2023. Yeah. So we are going to finalize the table or our presentation between February and March next year. Then we will present it, if possible. It depends on the information we receive, but if possible, we are going to publish article based on the information from each country's or regions.
Okay, thank you. That's it. Thank you for so comment and the answer.
Thank you.
Thank you very much for giving this opportunity.
Today, we reported our APACRM activity and the current progress for regulatory harmonization. Firstly, I would like to say thank you all the speakers, panelists, facilitator, participants on APACRM. In this year, on the working group 1 session, we discussed about the point of non-clinical assessment to initiate clinical trial for CAR T cell therapy product. On the working group 2 session, we discussed the requirement of biological ancillary materials that had a starting point for harmonization in the future. On the working group 3 session, we just have started the activity as running each other. We would like to discuss about regulatory requirement for specific reviews and environmental impact assessment, building a viral product setting in next. Anyway, it is important to share information and exchange opinion each other. We should build a common view on prospects and challenges for regenerative medicine. We are preparing to publish this result as a proceeding paper, as someone said, mentioned. Okay, I'm afraid our time is almost up. I want to thank you all for taking time out of your busy schedule to be here today. Thank you.